All right, good evening, everyone. We're going to get started here in just a couple of minutes with this installment of the Fall Master Beef webinar series. We're excited to have Dr. Jon Beever with us tonight. Dr. Beever is the director of the UTIA Genomics Center, and we're pretty excited to have him with us tonight to share a little bit about some gene editing. So once again, if you have questions throughout Dr. Beever's talk, go ahead and stick those in the Q&A box. And we'll address those at the end of his presentation. But at this time, I'll turn it over to Dr. Beever.
Okay. Thanks for the introduction. Let's see if I can figure this out. I think I can. Maybe. Is that title slide up?
Yep, looks good to me.
Okay. So thanks for the opportunity to present this. I think it's I don't know how important of a topic it is. It's a growing area in animal science and livestock production, and that's this area of gene editing. And so the title, the first part of the title is meant to be a teaser: designer genes, and then of course the potential of gene editing in livestock production. And I'll tell you that some of the things I'm going to show you don't necessarily fit into the brand new category. We've actually been conducting gene editing experiments in livestock for at least with the newest version of tools since probably 2010, 2011. And so what makes the first example I'm going to give you tonight interesting, well, it'd be the first or second cattle example, is that what I'll tell you is that the release of these species or these animals that are edited into the production environment is a tightly or highly governmentally, federally regulated process. And that process is regulated by the FDA. And like I said, I'll tell you a little bit more about that in a minute. But the point of it is that we're now at a kind of a breaking point where we think that these technologies could certainly benefit livestock production. Yet we are having problems negotiating these regulatory hurdles that we seem to be coming up with. And the example of the SLIC gene, which is one of the examples I'll show you tonight, is an example where that regulatory process was, I won't say it was sidestepped, but it gained approval due to some conditions that existed in the scientific literature at the time. And FDA approved it with hardly any regulatory documentation for use in livestock. So that's the new thing, and that was done just here this spring in April. So there are a lot of these things in the pipeline, and I'm going to go through a bunch of them today. But gene editing is real, and what you guys need to be thinking about is where it can be implemented, and how it can be implemented, and whether it's of value. But I think the eventual answer is, as we push toward our grand challenges of 2050, is it could be exceptionally valuable to the livestock industry. So without further ado, I'll get to it. Or maybe I'll get to it. There we go. So first, I think it's important to go through some definitions. There's a glossary at USDA that covers all this, and that's referenced in that URL down below on this slide. Gene editing or genome editing, either term is part of what we would call agricultural biotechnology. And by this definition, agricultural biotechnology is a range of tools, including traditional breeding techniques that alter living organisms or parts of organisms to make or modify products, improve plants or animals, or develop microorganisms for specific agricultural uses. And then a little tack on the in there, modern biotechnology today includes the tools of genetic engineering. and gene editing falls under genetic engineering. A little side note, for those of you that are aware, there is now a USDA certified label for genetically engineered products. And that is what you will begin to see as soon as some of these things begin to come to market, is that just like you have a USDA organic sticker or a USDA, I can't think of one offhand, like a USDA inspection or past federal inspection sticker, you will have to have a GE sticker, a USDA GE sticker on the label of these products before they can be put in a retail environment. But the G is going to stand, of course, for genetic engineering. So I think you guys know the definition of traditional breeding, but there's some nuances here that I want to point out. Traditional breeding, of course, is the modification of plants and animals through selective breeding. I think you've probably heard over the last year and a half, probably some presentation on genomic selection, either by Dr. Rowan or maybe 3 years ago by myself. But basically the use of some really modern technologies to actually make selection progress in our species more rapidly. What you have to understand when you think about selective breeding is you're selecting for particular variation within the DNA of that animal. So that animal becomes a selected individual because that individual has the genetic merit or has the DNA information that makes that animal perform better than his counterparts. And so we do need to realize that when we put selection pressure on our animal populations, we are actually changing the genetic makeup of those animals. But it's through a process that is naturally evolving and based on information that we collect as animal breeders. The little caveat here in this definition is that traditional plant breeding has for years been able to do things that would be perhaps tucked underneath the biotechnology label, in that they've been able to modify the genetics of plants in tissue culture, and then by something called mutational breeding. And mutational breeding is basically the radiation of seeds from either the radiation of seeds or the radiation of plants, and then planting those seeds and see what that radiation caused to mutate, and then seeing if it was advantageous or not. And so, we've kind of been, for lack of a better term, either through natural selection or some of these biotechnologies, we've actually been modifying the genome of our domestic animal species for thousands of years. And that's kind of the punchline here, is that what I'm going to talk to you about tonight is this kind of next stage of genome modification. And it may happen in the lab, but rest assured that it's tightly regulated process and could be quite beneficial. You got to think about it as there's going to be a topic here. I'm going to show you a YouTube video. I'm going to show you that where somebody corrects somebody and says that this is precision breeding. In other words, we can introduce mutations that cause phenotypic effects or changes in traits in one generation and then propagate that. And I'll show you a couple examples of that also today. So some more definitions: genetic modification, the production of heritable improvements in plants or animals for specific uses. And see the last point here, where genetic modification includes either traditional breeding methods or genetic engineering. And so in theory, by these definitions, all animals that we breed and that we selectively breed are in fact genetically modified, either through traditional selection programs and then in the future here through genetic engineering. So today we like to begin using the word genetic engineering or bioengineered. That's another term that we can use. And what this implies is the manipulation of the genes by making either introductions of new sequences, eliminating, rearranging specific genes, and basically using these modern molecular modern biology techniques to make changes in the DNA. And if we do this, then we like to refer to that as a genetically engineered organism. And this is favored in contrast to genetically modified because of the there's two issues with genetically modified, is because that can be achieved through traditional means. And back when we started doing some of this stuff in the 80s, it was highly frowned upon by consumers, and to a certain extent, it still has some negative connotations in the population at large. But these are slowly being resolved. I teach a class on animal agriculture and society, and we talk about consumer preferences. And there is a relatively small group of consumers that prefer to not consume GMO products or GEO products. But that percentage is getting lower and lower as more education gets out there as far as these technologies. I want to distinguish here. I'm going to give you the definition of this transgene or transgenic organism. This is 30-year-old technology or 40-year-old technology. And I want to make it clear that although this may still happen, if warranted or if thought to be advantageous, what I'm going to talk about today are not transgenic. This is simply going in and actually editing the DNA sequence of an animal by making a few changes here and there to derive a certain phenotype. And so transgenic is the transfer of genetic material from one organism to another using similar molecular biology techniques. And I'll show you the actual first animal that was FDA approved here in a minute. But it is actually a transgenic organism. I'm going to try to distinguish between a transgenic organism and a genetically engineered organism here in a minute. So for animals that are genetically engineered, like I said, the federal regulatory authority over that is the Food and Drug Administration, and that is derived from the FFDCA, the Federal Food, Drug, and Cosmetic Act. And that was enacted sometime in the late 70s, I believe. In that act, it defines drugs, which is the definition below, as articles intended for use in the diagnosis, cure, mitigation, treatment, or prevention of disease in man and other animals. And then what the red part is important is, and articles other than food intended to affect the structure or function of the body of man or other animals. So just an example, the slick example I'm going to talk to you about tonight. The intention is to derive an animal that has a much shorter hair coat than its contemporary individuals. And so we are indeed affecting the form or structure or function of that animal. And that's why FDA has regulatory authority over these technologies. On the plant side and on the microbe side, that authority actually lies with USDA and APHIS, the Animal and Plant Health Inspection Service. And there's a huge contrast there in that, if you look today, there's probably some 10,000 different modifications that exist in crop species, only a certain amount of which are used actually in production, but there's thousands of them that have been approved for use under APHIS and USDA approval. So it's turned out that the hurdle for genetic engineering of animals has been a much higher hurdle than it has been in crops and microbes for that matter. And it's because of the regulatory body being the FDA. Part of that problem is the standards that the FDA applies to any drug. One is it's safe, and in this case means after we modify it, can you eat it? And does it cause any harm? Does it cause any harm to the animal that you've modified? So in other words, does it compromise their health and welfare in any way? Is there potential for environmental damage due to the modification? And can the producer or the user raise these animals with safety? Effectiveness, does it do what you say it does? That's a measure of about every drug, not about every drug, but every drug that's approved. So, does it cure, whatever disease, yada, yada, yada. In animals, we ask, well, I've made this modification. It's supposed to increase disease resistance or shorten the hair coat or make an animal be born without horns. You got to prove that it does what you say it does. The quality of the product, that's a tough one, because then you say, well, they ask, how is it produced? Well, you know that in pharmaceutical factories, there's certain regulatory approvals and certain standards that need to be upheld. That's not something that we are necessarily interested in from an agricultural standpoint. And having our farms and our production facilities under regulatory control to where we have to produce animals in a particular way in certain conditions to ensure that the product is always the same. And so that's been part of the issue. And then properly labeled, believe it or not, that properly labeled has only been solved here in the last 1.5 or 2 years, and I'll show you why that's happened here in a brief second. Actually, right now. So what I'm going to tell you first, I'm going to tell you a story of a salmon, which is, I know, not a beef cow, but I'm going to tell you the story of a salmon called the Aqua Advantage salmon. And the Aqua Advantage salmon was produced by a company called Aqua Bounty Technologies. This is a picture of the paper that was published in a journal, believe it or not, 30 years ago. So 1991. And you say, well, I've never heard of that, or maybe I've heard of it, but I've never been able to eat any. Well, there's a reason, and that's been this regulatory process. This was an example of a true transgenic animal. What they did is they made a DNA molecule. We call that a construct. And that DNA molecule contains several different segments of DNA. One of those segments is this. I don't know if you can see this in the figure here under A. You have something labeled OP-AFP promoter. That is a DNA sequence that was derived from a ocean pout, which is a species of fish that grows in very, very, very cold waters. And that particular promoter is active when the fish is in cold water, and it actually encodes the protein actually encodes an antifreeze protein, so those animals can survive in very cold conditions. And the reason why they use this promoter is they wanted the growth hormone gene attached to it to be expressed during certain parts of the life cycle of Atlantic salmon, where they tend to grow slow in colder waters. They attach that promoter to a DNA sequence from coho salmon that encodes the growth hormone, and then they added some DNA on the end of it that was from that ocean pout antifreeze protein. And they inserted this DNA into the genome of the Atlantic salmon. And sure enough, it worked. If you look at these red boxes here, you can see that both from a growth hormone standpoint, they had increased concentrations of growth hormone compared to control fish. They had increased weight compared to control fish. So here you see that the weight, for instance, in October of 1990 was about 21 grams, where the sibling or control fish were 7.4 grams. And in January of 91, they weighed 47 grams, where the control fish weighed 9.5 grams roughly. Okay. So you can see the massive difference in growth here. Length, of course, is the same. It's related to overall body size, but these fish are quite a bit longer than their counterparts. And you can see the growth rate here. The growth rate is roughly, that's percent per day. So they were growing quite rapidly. This is what it looks like from a picture standpoint. These two fish are the same age. The small fish is a normal Atlantic salmon, and the large fish is a genetically modified or genetically engineered salmon that carries this DNA construct. And so upon documenting this through scientific literature and other measurements and performance metrics, Aqua Bounty almost 30 years ago attempted FDA approval of this fish, and it was just granted in 2017. Okay, so it was almost a 30-year process. The reason why you've probably not seen it in grocery stores is that as soon as the FDA approved it for commercial use, there was some politics that went on that prohibited it from being imported into the United States. So as soon as it gained regulatory approval, there was a law passed that it couldn't be imported into the States. And then there were a few other things that occurred along the way. One was a labeling issue, which again has now been resolved. And so I suspect that you will be able to see this product in grocery stores sometime within the next year. If you're in a certain market, you might still be able to see it now. But like here in Knoxville, you might not be able to see it for another year or so, but I think you'll finally be able to see that. And I'd like to show you a video. Let's see if I can do a new share here. I'm going to show you a video of this company, and what I want you to think about when this video is playing is the impact that this technology has from our grand challenges standpoint, is the ability to feed people, the ability to utilize resources efficiently, words like sustainability, things like that. I want to show you what this company's up to and how this might work in regular livestock species. So let me see if I can do this seamlessly. Evidently not really. So this is like a 5-minute video.
Here at Aqua Bounty, we're pioneering a sustainable and nutritious future by transforming aquaculture. With a growing global population and increased consumer demand, the existing supply of seafood can't fill the gap. We know we can't fish our way out of this. That's because catching more wild salmon would potentially harm wild fish populations. We also know that building new sea cage salmon farms can have a problematic impact on the oceanic environment. Aqua Bounty is an innovative solution to address growing supply and environmental concerns. The Atlantic salmon bred by Aqua Bounty is a safe, secure, and sustainable alternative for anyone looking for fresh salmon that is good for them and good for the planet. So here's how it works. Aqua Bounty Atlantic salmon were genetically modified one time 30 years ago to help the fish during the critical early stage of growth. Aqua Bounty raises their salmon in carefully monitored indoor land-based fish farms in North America, where the salmon swim and school naturally in a way that conserves water and other natural resources. Our freshwater farms are built to grow and deliver fresh, nutritious salmon that is free of antibiotics and other contaminants and with no impact to wild fisheries. Aqua Bounty's genetically engineered Atlantic salmon eggs are produced using the same methods as conventional salmon farming operations, with one primary difference. Aqua Bounty salmon eggs are sterile, so that fish from these eggs are unable to mate. The process starts in our broodstock facility on Prince Edward Island in Canada. That's where we maintain our male and female broodstock, which are carefully selected to produce the highest quality eggs. The female fish come from superior lines of conventional Atlantic salmon, while the males carry the genetically engineered transgene. This GE transgene is a permanent part of the male's genetic makeup, which was inherited naturally from one generation to the next through conventional breeding over 3 decades on Prince Edward Island. When spawning and fertilization is completed, the sterile all-female GE salmon eggs are placed in incubators until they develop into eyed eggs. These eyed eggs are now ready for shipping and are safely transported to our farm in Albany, Indiana. Upon arrival in Indiana, the eggs are transferred to the hatchery in our farm, where they are incubated until the eggs hatch as tiny fry. Soon after hatching, the fry are moved into small tanks in our nursery room, where they begin to actively swim and feed on a special starter diet that help them thrive. As the fish grow and develop, they need more room. When the juvenile fish reach 50 to 100 grams, the fish population in a tank are divided and transferred into larger tanks in our pre-grow out room. When they reach 500 grams, they are again divided and transferred to our largest tanks in the grow out rooms. Here they continue to thrive and develop into market-sized salmon, weighing approximately 4 to 5 kilograms or 9 to 11 pounds. All Aqua Bounty's GE salmon eggs and fish are reared in closed buildings with strict biosecurity measures. Netting, screens, and filters are incorporated into the farm design as multiple redundancy to prevent escape. Aqua Bounty's farms utilize a recirculating aquaculture system. Only fresh water is used from hatch to harvest in our farms, and this water is continuously recirculated. The process features biofilters, which remove waste and returns fresh water to the tanks to maintain an optimal growing environment for the salmon. Over 95% of the water in the system is recycled, conserving our freshwater resources. And our system allows for an additional benefit for traditional agricultural farmers. We continue to build partnerships with local farmers who utilize all aspects of the fish, like the waste that is removed from the water as fertilizer. Creating jobs and supporting farmers in the U.S. heartland is important to Aqua Bounty and an important aspect of our commitment to rejuvenate rural America. When it is time to harvest, our salmon are transferred from our grow-out room to our conditioning room. Feeding is stopped and salmon continue to swim in freshwater until the point of harvest. Our processing team treats the salmon with the greatest care and attention to maintain the highest quality of our final product. In all, it's an 18-month journey from egg hatch to harvest, and we don't say it's complete until we've delivered our healthy, tasty, and sustainable salmon to our customers. Aqua Bounty is thinking big, working to make our world a better place using smart science and safe, proven technology to solve the serious global problems like food security and climate change. As thought leaders in aquaculture, Aqua Bounty is helping to transform the future of seafood.
So, whoops. So a couple of things to point out throughout that video is one, think about this in the context of other species, the other livestock species, and being able to follow along the same kind of model to accomplish what these guys have accomplished. I think it's possible. I think that's the way we need to be thinking as livestock industry. The second part is they mentioned the biosecurity and the everything's being enclosed to prevent escape. One of the major concerns was the escape of these animals into the wild population. What would happen if this transgene was passed along in the wild population? And in order to circumvent that regulatory issue from the EPA, they made the fish sterile and they moved to completely confined production. But you can see that it works. I heard something, I think last year they produced some 50,000 tons of salmon in their facilities. And like I said, I expect you'll be able to see it soon in your grocery store if you're a salmon lover. But again, this was the old technology, and now we're going to move on and talk about some new technology. Let's see, I hope I'm sharing that right, but it's this just a second here. Let me stop this and now I've lost you guys on Zoom. So new technologies in what we call this genome editing have been around or were developed, started probably in 2010, 2011. And there are three basic technologies. I suspect that you probably have not heard of the ones called zinc finger nucleases, or the one called transcription activator like effector nucleases, or TALENs. But I suspect that maybe in the popular press you've heard several times, at least in the not too distant future, or not too distant past, this term called CRISPR. And CRISPR stands for clustered regularly interspaced short palindromic repeats. And I'll tell you what that is here in a second. But the bottom line of these technologies is that they are all tools, or they're all techniques that are directed at making breaks or nicks in the DNA molecule. And then what happens when you make a break or a nick in the DNA molecule, the cellular repair machinery are recruited to that site to repair that break. And during that repair process, we can actually leverage that to introduce mutations at a very precise location. And in fact, we can repair, or when we repair, we can simply change one nucleotide, and I'll show you some examples of that in the following slides. But the bottom line here is that these are tools made to target very specific DNA sequences, and then after targeting those sequences, make DNA breaks in the DNA or nicks in the DNA to elicit the cellular machinery for repair. And then we hijack that repair mechanism to actually make these modifications. So CRISPR-Cas has emerged as the tool of choice. It's actually a naturally occurring biological process that's derived from some microbial systems. It actually acts as a primitive, which may be spelled wrong there, a primitive immune system. And basically, whenever some of these microbes get infected by their pathogens, which are typically viruses, they wind up storing away a little piece of DNA from those viruses, and then continually express those pieces of DNA in order to cut up any new viruses that they come in contact with, or versions of the old virus they come in contact with. And so what we've done is we've hijacked that system. There are two parts of the system. There's this Cas part, which is actually what does the break in the DNA, or cuts the DNA molecule. But then there's this pink part here is referred to as a guide RNA. And that guide RNA is a very small piece of RNA, about 20 nucleotides in length, that you design to be complementary to a specific place in the genome of the organism you want to modify. They're very small molecules. Like I said, they're only 20 nucleotides. They are synthetically manufactured. I can go online here and I can order one in about 5 minutes. They're very easy to use. So all of these have been commercialized. This Cas9 enzyme that cuts the DNA is commercially available. This looped RNA, which is part of that Cas9 associated part, can be commercially bought. And then, like I said, the pink part that targets a particular region of the genome can be purchased. And so you can combine these three things together, put them into cells, and while they are in the cells, they will actually target the specific spot and break the DNA. And then we can hijack that to either change the DNA sequence or potentially even insert some sequence, etc. They're also very cost effective. I can order all these reagents for probably about $600 or $700. And then begin to modify cells at targeted locations I want to target. So to talk to you about what the tease was, the slick locus, let me tell you how we decide to do what we do from a gene editing standpoint. This is a paper from 2014, and you see the title here: Functionally Reciprocal Mutations of the Prolactin Signaling Pathway Define Hairy and Slick Cattle. And this figure is directly out of that paper by Littlejohn et al. What's shown here in this image is two animals that have differences in the DNA sequence for actually the prolactin gene. So the wild type animal, which is on the left, actually has a normal prolactin gene, and the mutant on the right has an abnormal prolactin gene. And you can see the difference in the hair coat. So that actually makes a shaggy phenotype. There are a number of issues with that. These animals also do not milk well. They have some reproductive issues. And so this was actually a mutation that was selected against. And so, but you can see what the addition of hair does to an animal. You can see that those that have the mutation that make them shaggy have elevated body temperatures under different ambient temperatures. And so this is definitely not a good thing, not only for the hair coat part, but for the for milk production and fertility also. The second mutation they found in this paper was a mutation that exists in Senepol cattle. Senepol cattle were, for those of you that are unfamiliar, were a derived breed for heat tolerance. And so what was noticed in the population of Senepol cattle after it was developed was there were some of these animals that were very slick-hided. And so you can see here this animal on the left-hand side, barely any hair in the ears, barely any hair on the poll. And of course, the hair throughout the body is very short and the animal appears very slick. On the right is a Senepol animal that does not carry the genetic mutation causing the slick-haired phenotype. This mutation is actually a mutation in the prolactin receptor. And so that title what that title was trying to indicate is that if you mutate either prolactin or its receptor, you can get these changes in hair phenotype, and in particular in this case, and potentially other phenotypes also. So the prolactin mutation was negative, not only from a hair coat standpoint, but from a productivity standpoint. The prolactin receptor mutation does not appear to have any negative impact from milk production and fertility standpoint, and has a positive impact from shedding and slick hair coat. And so you say, well, I want to have slick-hided cattle, and I'll show you some more data here in a second as to why you want to do that. So how do I get this slick-hided phenotype in my cattle? Well, turns out we have an answer to that. This is a paper that was published in 2014 also, very close after the previous paper I talked about. This actually comes from University of Florida. Back in the 1980s, the story goes that University of Florida was donated some semen for breeding their Holstein cows at their dairy. And like I said, as the story goes, I don't know if the story was made up in hindsight or not, but there was also some Senepol semen in that tank. And in fact, they used some of that Senepol semen to breed their Holstein cows. And then from that point on, graded up back to 15/16 Holstein while selecting for milk production, et cetera, and selecting for the slick phenotype. Now, at the time, they had no idea the mutation involved in the slick phenotype. They simply knew that these cattle were slick. And this is actually a production paper. And so here's some graphics from that paper. These are measurements of vaginal temperature. So the tool they used for this is they took a cedar and put a thermometer on it and inserted it into the groups of cows, and then measured vaginal temperature throughout the day. And so you can see as they plot it here, down over here on the left-hand side is 2300, which is of course 11 p.m. And then they go through their day, and you can see the fluctuations in body temperature. This curve on the bottom here is actually slick cattle that carry the mutation or were slick-hided. and the curve on the top is all the other cattle. This big dip here around 8 a.m., they justify that by indicating that that was feeding time, and immediately following feeding time, these cows would go out and then drink a huge quantity of water, and that would cool their body temperature for a little while, and then it would return to normal. Either way, what you see here is you see almost a full degree somewhere between a half and full degree difference in body temperature throughout the day of these cows. And so this is just a normal production environment. Same thing here. If you actually look at rectal temperature, respiration rate, skin temperature, and sweating rate, you can see that these cattle that are slick outperform the other cattle in these groups. And so here in the upper right or upper left, you see that the rectal temperature of the cattle does not rise as much during the day as a normal animal. On the lower left, you see skin temperature, which is a little bit strange. So you see the first part of the day, the skin temperature rises, but then after about 11 a.m., you see the slick cattle begin to decline. It turns out that the slick cattle have better sweat capacity too. And that's seen in the bottom right-hand side, where the corresponding temperature curve and the corresponding sweating rate are in the sweating rate, it levels out for the slick cattle, and it decreases its body temperature for the slick cattle on the bottom left. In the upper right, this is simply respiration rate, and as animals are not under heat stress or at a normal temperature, they're going to maintain their respiratory rate, and so of course you see these slick cattle do that quite nicely. These are dairy cattle. I know the metric we use for dairy cattle production is actually milk production, which of course is important in beef cattle also. This is actually a graph of milk production, and the two lines in each of these graphs, the open circle and closed circle, are actually based on calving date. So the black dots in this top graph are cows that calved in the wintertime, and then were measured for the first 90 days of milk production for their milk production. And the cows here plotted with the open circles were calved in late spring. In other words, during that first 90 days, they were going to be exposed to heat increase. And you see the impact of that heat increase on daily milk production. Almost 5 kilos or 6 kilos difference between cool season milk production and warm season milk production. So of course, that's a production loss. The slick cattle, although there's a lot more variation you see here, they still maintain a higher level of production and equal level of production for both seasons. So, again, so here we are looking at a direct production advantage for having slick hide. Okay, so the punchline here is that we see the benefit of having slick hide. What I didn't tell you is in order to get from that first Centipole cross to a productive Holstein, it took almost 35 years to get back to Holstein production levels. So I'm not sure that, so we could integress, the point is we could integress this mutation into any population of cattle. In fact, it has been integrated in a few, as well as a few other mutations that have been found since then. But what if we could just move this mutation into a population of animals directly without all the downtime? And so that's what happened this past spring. You see the title here, US FDA clears the way for CRISPR beef cows, CRISPR being the modification method, but basically the slick coat phenotype was produced by editing the prolactin receptor gene in a very targeted way. This is just a picture of some Centipole cattle, and you see, of course, that they're slick-hided. I have, I'm not going to play all of it, but I have a thing from Wall Street Journal that I'll start playing, and then we'll shut it off here. See if I can get to it again. Oops.
Meet Genzel, a 2-month-old Angus calf that could signal the dawn of a new era. Gene-edited animals designed by humans for human consumption. First in the world. First in the world. The company that made her says she's born with traits spliced into her DNA that should allow a cold weather cow to thrive here in the Brazilian tropics.
And what we're seeing now is that it works.
You're about to find out why, if indeed this works, our world is about to change, and why the economic incentives driving gene editing technology are so powerful. With 200 million people, Brazil is a vast country with a vast appetite for beef. That's because the US is the top producer of Angus meat. Most of Brazil's meat comes from zebu. Do you think the regulators here are open to gene-edited meats?
I think so, yeah. The agricultural sector is very open and keen of new technologies.
Fernando Flores is one of the lead scientists at the Brazilian government's.
So I'm going to stop it there just because it becomes a little bit of an advertisement for Excelogen. When he said it's the very first, he was talking about the very first to have this slick mutation introduced. I'm going to show you examples that go all the way back to 2010, where gene editing has been done in different livestock species. So, but the point being here is, and those of you that might debate that they keep calling that Angus, and of course it's a red Angus, but the point being is that it can be introduced in any breed, and they've done it. They did it in Brazil because of the regulatory problems here in the United States, because Brazil was receptive to having those cattle there. But now FDA has approved them here, so you'll see them eventually, but it'll take a little while to get more of those on the ground. So anyway, we'll get back to our slick cattle. That link should be active. I'm going to go ahead and give this presentation to you guys. That's where you can access it, but that link should be active. But I want to show you some other examples of things that have already been done, and they've been done for quite a while. This is actually the modification of a gene called myostatin, and they chose to do it in the lower cattle. And the reason why they chose to do it is because you can see that the muscularity or the muscle type in these cattle is not, it wouldn't be what we consider here in the United States very good. These are two calves that were born following gene editing of the myostatin gene. And this heifer calf is not edited, and the bull calf is edited. And I know it's a difference between a female and a male, but the bottom line here is these are when they're yearlings, and you can see the lack of muscularity in the heifer versus the increased muscularity in the lower bull here. And so that was actually done by the same company, Excelogen, but it was also done in Brazil. This little guy over here standing up on the upside down feeder has also got a mutation in myostatin. And what I think they were trying to show you is the hip structure on this lamb. And of course, he's got more muscle in him also. These are the mutations that were introduced. Basically, the removal of 3 base pairs in the myostatin gene. Here, the removal of 1 base pair in the myostatin gene. And then you see that the female, of course, was completely normal. There was another bull calf born that had 3 base pairs missing in the myostatin gene. He died. And then there was another one that had several different mutations. So when we target these regions, so the target area was in here. And you can see the mutations were actually made where they belonged. This one is a little bit more extensive. It had a little bit larger deletion of DNA than was desired. The sheep, same way. The sheep winds up being heterozygous, and actually carried the same mutation as the bull, and that's not necessarily by coincidence. the repair mechanisms when they go to repair the DNA molecule use certain cues to make the repair. Evidently the absence of these 3 nucleotides is somewhat allowed, and the repairs happen the same exact way. But simply what we're doing is we're going in, targeting a DNA sequences, manipulating that DNA in a certain way, and then hopefully the result is what we desire. Here's another one. Of course, those of you that know cattle know that almost all the dairy breeds are horned, and dehorning is a huge animal welfare issue to some people. And so the idea being here is, can we make a polled Holstein? And here's the first paper that described that. And that was done in 2016. This bull here actually went on to successfully produce offspring that were, of course, polled. And so he was mated to a number of different horned cows. And of course, the offspring all came out polled, like you would expect under the dominant inheritance pattern of polled. And so what they did in this paper here was insert the mutation that is commonly found in Angus cattle into these Holstein embryos, and then produced live offspring. And those live offspring were fertile and able to pass that mutation on to their offspring producing polled cattle. So that's a long way off from being incorporated in today's modern Holstein genetics. But the point being is that it's possible to do. And this was actually the insertion of about 300 nucleotides into the DNA sequence that actually causes polled. Here's another one that I had the opportunity to work on a while back in 2016. Mannheimia hemolytica, you guys all probably vaccinate for it. It's one of the most common respiratory disease bacteria that causes most problem. It doesn't necessarily cause a problem, the bacteria itself, but the bacteria produces a leukotoxin that then does some significant damage to the white blood cells that migrate to the location of the infection, and causes even a worse pathology. And so the thought here is that if you could make the leukocytes of the cattle resistant to this leukotoxin, that it could actually solve the disease problem with Mannheimia hemolytica. That has been demonstrated in this PNAS paper. And in addition, following the publication of the PNAS paper, there was a group that went out and looked at many thousands of animals, trying to identify animals that may be naturally resistant to leukotoxin. And they didn't find any of those, but they found some that were more susceptible. And again, caused by changes in the DNA sequence of this gene called CD18. So you make a certain mutation in CD18 and you get resistant cattle. You make other mutations in the gene and you potentially get cattle that are more susceptible to leukotoxin or susceptible to BRSV, or sorry, bovine respiratory syndrome, not bovine respiratory syndrome with syncytial virus. Oh, here I am not doing this in presentation mode. PRRS is another condition in livestock industry and pigs that cause significant losses, somewhere between 600 million and $1 billion per year in the United States. Turns out that if you mutate a gene called CD163, that makes the pigs completely protected from PRRS virus. You can put PRRS virus in their nose and they don't get it. This is as close to commercial production as any of these technologies in livestock species. Recently, here in September 21, there was an agreement made between Genus, which of course is a genetics company that has worldwide impact from a genetic selection standpoint. They have pigs, cattle, and chickens. Cattle through their ABS genus arm, but they've made an agreement with Roslin They licensed this technology and they made agreement with Roslin in Edinburgh, Scotland to produce these pigs for future measurements and FDA approval. So they've licensed the technology. They're generating data for FDA to approve them. And I suspect at some point in time, you will be able to purchase PRRS resistant pig germplasm. I suspect probably sometime in the next 2 to 3 years, probably. Here's an example that a colleague of mine at Illinois and I did 7 years ago now, 8 years ago now. This just shows you exactly how we might do it. There are other ways to get it accomplished today, but back in 2013, we did it this way. We isolated skin cells from a fetal pig, or primary fibroblasts from a fetal pig. We treated those cells with a zinc finger nuclease, and then cultured those cells for a few days, sorted them into individual cells, and then tested those individual cells for the presence of mutations. And this is kind of a insight to the efficiency of that process, which is no longer a problem, I'll say. This is the normal sequence. This is the sequence here that we targeted in the underlined by the dots. And you can see that we targeted that position, in fact, derived a number of different mutations, whether it be the subtraction of a T, the addition of a T, the addition of 2 Ts. But the punchline is here. We screened almost 3,000 cells to see 32 that had been modified. The efficiency of CRISPR technology today is much, much higher than that. CRISPR will generate modifications in that 20 to 50% range. And so this was like looking for needles in a haystack, but nonetheless, we actually produced pigs that had this mutation, and we just published this paper here this summer. We only produced female pigs, and then we bred them for a number of generations. These are littermate pigs. The pig on the left has the mutation we introduced into myostatin. The pig on the right is a normal sibling that does not have the mutation. And I think it's fairly obvious that you can see the difference there. That the loin eye, these pigs weighed exactly the same amount when they hung on the rail, 301 pounds carcass weight. They were a little bit big, but they weighed exactly the same. And you can see the difference in lean to fat ratio between these two carcasses. And so that's just the addition of one mutation in the myostatin gene can have that impact. Now, the other part of that story is that when you breed them to homozygosity, or you make them have 2 copies of that gene, they die after 24 hours. So you will probably never see this one on the grocery store shelves either, because it violates the rule of animal well-being if you breed them the wrong way. This is another project that we're actually doing in my lab currently. This involves 2 different genes. These are mice. Of course, mice breed a lot faster than cows, but these are mice. This is a normal mouse on the very far left. The second mouse from the left is a mouse where we've introduced a mutation in a gene called IGF2. The third mouse is a mouse where we've introduced a mutation in this myostatin gene. And then the fourth mouse, the one on your far right, is the one where we've actually gotten both mutations in there. And the difference in weight between the one on the far left and the one on the far right is 100% difference in weight. So the one on the far left weighs about 20 grams live, and the one on the far right weighs about 40 grams live. And so we can begin to stack these introduced mutations into animals to see if we can increase productivity. Currently in the lab, we are actually modifying a cell line to have both of these mutations in cattle. And we hope that within the next year or so, we'll have a live calf born that carries both these mutations and in theory has the same benefit that we see here in the mice. That's all I have. And so now we can have all kinds of questions if you want. I don't know what time it is, but.
So far, it looks like we only have one question. Someone said they couldn't see the info on the cattle. How can I get more information on genetic modification?
I'm not sure I understand the question. So you can, if you search the term genome editing and either put cattle or livestock, you can pull up information on about any of the examples I gave you.
Okay, we had another question come in. Says, Dr. Beaver, I'm not a Luddite and recognize that this technology has left the station and is coming. You mentioned, and the video also stated, that biosecurity is carefully maintained. If the GME animals are safe to eat, why are they not safe to reproduce naturally?
They are. It's just going to be getting through that regulatory process. Like I said, FDA has tended to treat these things as drugs. And so when they talk about production facilities, they're going to have to, I think we're going to have to convince them to kind of bend their minds in a way where a drug manufacturer and a beef producer are not the same thing. And we're going to have to prove that. So there are no wild cattle in the United States. So, you know, if your cow gets out and breeds the neighbor's cow, well, that may be something, but there are no wild cattle populations to impact. You know, many of these mutations that we might be attempting to put into the cattle, you know, the upsides are, much greater than the downsides. And again, as long as we're paying attention to potential environmental impact and animal health and welfare and making sure that that's up to snuff, most of the mutations that are being introduced do not change the product at all. And so on those terms, we believe that the FDA will kind of get in the game here and allow us to produce these animals under normal production practices, and not, you know, not have the regulatory burden of, you know, inspection and things like that.
Looks like we've had another
Yep.
question.
Yeah, I
Okay.
got that one. So maybe. I think that the distinction here is I mentioned Genus had licensed the IP for the PRRS resistant pig. They have actually also licensed the IP for the Mannheimia hemolytica resistant cattle. I think the cost of those cattle, or the upcharge on those cattle, will be reflective of whatever it costs them to get through that regulatory process with FDA. But downstream, I'm not sure that the incremental cost increase is going to be that much. I can't say that for sure, but you're correct. There is that potential, but I think there are enough academic units out there that'll be balanced. That'll be balanced so that the increase in cost is not as much as what you might think. It's maybe a little bit different than seed corn or seed beans or what have you, where it's an extra $100 a bag. I don't know that that'll be the case. It might be a $3 upcharge on a straw semen or something.
Very good. Looks like we don't have any more questions right now. We'll give it just a couple more minutes to see if any last minute questions come through before we dismiss.
I guess part of the message here is that one, we don't actually try to modify anything unless we know what it does, or if we see a potential upside for it in the industry. And so, you know, that title slide of designer genes, I've said this for several years now, and I've given a couple talks, industry talks, is, you know, think about what convenience traits or what production traits you guys need to have in your cattle to make a difference, from your, from an economic standpoint or production standpoint. If we understand the biology behind that phenotype, which we're learning more and more and more about every day, the potential for editing to improve those animals certainly exists. And I just gave you a few examples. That paper that I showed you about the myostatin pigs is actually a review paper, and there's probably 26 references within that paper on previous attempts to modify growth promotion genes in goats, sheep, rabbits, cattle, pigs, etc. And that's just lean growth production. That's nothing disease resistance, or, there's a couple of examples out there. Cashmere production. There's a couple of groups that have modified a gene called FGF5 that improves cashmere production by 20%. So if you're a cashmere farmer somewhere where they raise cashmere goats, your bottom line increases by 20-25% with the cashmere you can harvest. So if you can think about what it is you want, we can try to figure out what it is we need to change.
So far, no last minute questions. So I think we're good to dismiss for the evening. Dr. Beever, thank you. We really appreciate your time and your knowledge on gene editing. We really appreciate it. Someone in the chat box is also saying thank you.
Yep.
So I think that's it for tonight. Once again, don't forget to take that survey that'll pop up on your screens once we end for this evening. And we'll see you next time. Thanks, Dr. Beever. We appreciate it.
Sure enough. Thank you.